Yes. Good afternoon, everyone, and welcome to you all. And a special and warm welcome to our Ph.D. candidate, Malan Johansen, and her family and friends. Today, as you know, Malan will have a public defense of her dissertation. It will be Epidemiology and Genetics of the Amyotrophic, Lateral Sclerosis, and Alzheimer disease in the Faroe Islands. The principal supervisor is Associate Professor Maria Skålum-Petersen, Department of Occupational Medicine and Public Health, and Faculty of Health Science at the university here in the Faroe Islands. She also has two co-supervisors, Professor Torben Hansen, the Novo Nordisk Foundation Center for Basic Metabolic Research, Faculty of Health and Medical Science, University of Copenhagen, and Professor Anders Albrechtsen, Section for Computational and RNA Biology, the Department of Biology, also from the University of Copenhagen. So those were the supervisors, and the assessment committee appointed by the university here in the Faroe Islands is Orla Heidemann, Professor of Neurology, Head of Academic, Clinical Medicine, Trinity Institution of Neuroscience, Trinity College in Dublin. And Jørgen Erik Nielsen, Professor of Neurology and Research Director of the Neurogenetic Clinic and Research Lab at Rigshospitalet in Copenhagen. And at last, myself, I'm Jon Ruth-Helling. I'm Associate Professor in the Molecular Biology at the Faculty of Science and Technology here at the University of Faroe Islands. So that was the name dropping. Now I will take you briefly through the agenda for today. In a moment, Malan Johansen will present her dissertation. That will take about 30 minutes. And afterward, the first examiner, Professor Orla Heidemann, will make her comments and ask questions. And then we will have a coffee break for 20 minutes. And the second examiner, Professor Jørgen Erik Nielsen, will make his comments and ask questions. And then there will be an opportunity for persons here in the auditorium to ask questions. And finally, we in the assessment committee will step outside for the voting, and we will come back with our final recommendation. So, before I give the floor to Malan, I will, as chair of the assessment committee, first present why the work of the Ph.D. candidate has been approved for public defense today. The thesis of Malan Johansen is article-based, and successful peer-reviewed publications reflects the quality of the work. The overall aim of the thesis is to explore the epidemiology and genetic susceptibility of ALS and the genetics of Alzheimer disease in the Faroese population. The scientific questions asked are valid. The hypotheses and aims are clearly enunciated, and the description of the background and scientific method is clear, adequate, and concise. There is a good coherence between the four studies, and overall the results and discussion are adequate. The publications of 3 papers and the quality of the manuscript in preparation support the observation that the work has made a contribution to our understanding of the genetic epidemiology of ALS and Alzheimer disease in the Faroese population as an isolate. The studies are carefully performed, and the structure of the thesis meets a high standard. The scientific writing is very clear and of very high quality. So, we all 3 members of the assessment committee find the dissertation to be of sufficient academic standard for public defense. And that is what is next on the agenda. So please welcome Malan, and the floor is yours.
Thank you, Jon. Okay, so I would also like to begin with welcoming the assessment committee and also my supervisors, and also a very warm welcome to everyone online and all of you here in the room, especially colleagues, friends, and family. And yeah, my thesis is titled Epidemiology and Genetics of Amyotrophic Lateral Sclerosis and Alzheimer's Disease in the Faroe Islands. And the presentation I will give you here today summarizes my 3 years of doctoral studies. The presentation is based on 4 scientific papers, of which 3 have been published. So I have just outlined the agenda very simplistically here, but as I have been working with both ALS and Alzheimer's disease, I will divide my presentation in 2, and I'll first talk a little bit about ALS and the 3 studies pertaining to ALS, and in the second part, I'll talk a little bit about background knowledge on Alzheimer's disease and the 1 study which I performed in relation to Alzheimer's. So, ALS. ALS is a neurodegenerative disease, and in ALS there are primarily 2 types of neurons affected, and these are the upper motor neurons and the downstream lower motor neurons, which are found in the motor cortex of the brain, brainstem, and the spinal cord. And these neurons are the ones that innervate skeletal muscle. And as these neurons die, this leads to muscle wasting and ultimately paralysis. As no effective cure exists to stop or slow the progression of Alzheimer's disease, death will typically occur some 2 to 5 years after the first symptoms. And many studies have sought to identify risk factors associated with ALS. However, the only ones that have been established are the ones listed here. And age is a risk factor for ALS. The peak age is somewhere between 60 and maybe 75 years at the peak age of onset. And also ALS is more common in men compared to women. And also family history is a risk factor for ALS, in that in 5 to 10% of cases, ALS seems to segregate within families. The remainder of cases are called sporadic. However, genes are also causal of sporadic ALS. So there definitely is a genetic composition to sporadic ALS also. The most commonly mutated genes are C9orf72, SOD1, TARDBP, and FUS. But genetic risk isn't really uniform across the world and varies with genetic ancestry. In European populations, the C9orf72 expansion is the most commonly mutated genes, as can be seen in the figure here. It accounts for approximately 30 to 40% of mutations in familial ALS and to a little less degree in sporadic ALS. But in other populations, the C9orf72 mutations aren't as common. And as an example here is the Japanese, where mutations in FUS and SOD1 are more common. So the genetic risk of Alzheimer's varies on a continental and global scale, but it also varies more locally. The allele frequencies may vary in population isolates also. And in the same way, the frequency of ALS varies across the world. Worldwide incidences range somewhere between 0.6 and 3.8 per 100,000 person years. And generally, the incidences are higher in Caucasian populations and somewhat lower in, for example, Asian populations. The incidence has been estimated in the Faroe Islands before by neurologist Malan Johansen, who began the prospective registration of ALS patients in 1987. And in his study, he found that the incidence during 1987 to 2009 was 2.6 per 100,000. And this estimate fitted quite well within European estimates. However, Johansen also observed that many of the patients diagnosed with ALS seem to originate from Føroya, illustrated here on the figure. Føroya, which is the southernmost island in the Faroe Islands. And this finding was indicative of a geographic cluster of ALS in the Faroe Islands. And shortly about the population, it today consists of approximately 54,000 inhabitants, and the islands were settled some 1,000 years ago by migrants from the British Isles and what we now know as Norway, mainly. And due to founding and subsequent drift, which is the random fluctuation of alleles, and also high rates of consanguinity, the Faroese gene pool is quite distinct compared to more outbred populations. And these effects have led to reduced levels of genetic diversity and increased frequency of some rare variants, and also increased homozygosity. And these are effects that may be beneficial when conducting genetic research. So given the observed geographic concentration of cases, the overall aim on my studies on ALS was to identify the possible cause of this cluster. And about the data, the Faroese Alzheimer's study included in 2020 55 patients of Faroese ancestry who were diagnosed with ALS. And at the time of the study, most patients were deceased. So information has been collected retrospectively from medical records. So information was quite limited, but we could find information on date of birth, date of disease onset, sex, and date of death and site of onset. And in the following, I will present the results of the 3 studies. First, manuscript 2, where the main aim was to provide an updated incidence estimate of ALS in the Faroes during the time period 2010 and 2020. As I mentioned, it had previously been estimated to 2.6 by Paul Johansen. So this study included 27 patients of Faroese ancestry. And we calculated the incidence using the person-year method. And we calculated accrued incidence, but also stratified by age group and sex. And also the incidence was standardized to the US 2010 population to enable comparisons with many other studies on incidence of ALS. We found that the accrued incidence in the Faroes was 4.9 per 100,000 person years, which was higher than the previous estimate. However, when standardizing to the US census population, the incidence was a bit lower. It was 4.1 compared to 2.9 in the previous estimate. And as seen in the figure, with age groups on the x-axis and incidence on the y, where we compared the 2 incidences for the 2 time periods, it seemed that the largest difference was observed in the oldest age group, between in the age group 65 to 74 years and 75 plus years. We also observed that the largest increase was in women, as can be illustrated in the figure here. So women are colored in red, and the previous time period is the solid line, while the dotted line is the new estimate. And as can be seen, the incidence seemed higher in women also. So all around, the incidence seemed higher, but caution should be taken when interpreting results from small-scale studies, which are prone to more uncertainty. In the next study, we wanted to examine if ALS clustered within families by estimating the frequency of familial ALS, and also compare the frequency of ALS across geographic areas. In this study, 43 patients were included, and what we did was to divide the Faroe Islands. To assess geographic clustering, we divided the Faroe Islands into 6 geographic regions based on historical geographic boundaries, and then we assigned a patient to a geographic area based on the birthplace of their mother. And also, through the genetic biobank, we used the Faroese genealogy registry to retrieve pedigrees of ALS patients, and we used these pedigrees to identify familial relationships between ALS patients. So we then estimated the frequency of familial ALS in the Faroes using 2 stringencies. We used a more strict criteria, where we included patients with definite familial ALS or probable familial ALS. These were the patients who had either a first or second degree relative with ALS or more. We also made a more lenient definition to include patients who we observed had more distant relatives with ALS. These had possible ALS, familial ALS. So we found that the observed cluster of ALS in Føroya was significant, and also, as illustrated here in the red box, the frequency of ALS was approximately 3 times higher in Føroya compared to the rest of the country, and this was a significant relationship. So we identified no patients with definite ALS, 6 patients with probable familial ALS, and 6 patients with possible familial ALS. So under the first definition, which was the more strict definition, 6 patients were identified with possible familial ALS, probable, and that gave a frequency of 14%. And we saw that 4 of these patients out of 6 originated from Føroya. Under the more lenient definition, 12 patients were identified, and the frequency of familial ALS was then estimated to be 28%, and half of the patients were from Føroya. And just to illustrate the pedigrees here on the right are the patients with possible familial ALS circled with red. There was one pedigree, the first here, in which 3 second cousins were identified, and in the other pedigrees, patients with the possible familial ALS were related also to patients with probable familial ALS. So from this study, it seemed that the frequency of familial ALS was relatively high. And this led us to the third study and final study on ALS, in which the aim was to examine the genetic underpinnings of ALS here in the Faroes. We were able to identify DNA from 20 samples of the 55 total. And 1 patient participated upon consent, but the remaining patients, or most of the patients, they were deceased at the time of study. So the remaining DNA samples were recovered, there were banked samples that we recovered, with DNA coming either from blood or, in one instance, dried blood spots from filled paper, and in 9 instances from formalin-fixed tissue. So this wasn't really an ideal situation, and unfortunately, the quality of the DNA from blood spots and tissue, which counted half of the samples, was poor, being degraded and fragmented. However, we went on with the studies, rather have some results with the material that we had than no information at all. And we could also use the DNA or the samples with poor quality to substantiate the results from the others. DNA was sequenced at the facilities of co-supervisors and also processed there. And the figure on the right shows the basic processes of whole genome sequencing. And these steps I had help with, and I analyzed the prepared sequence information. We performed short read sequencing with 30x coverage, and after quality control procedures, the effective depth of DNA was for blood samples 25 and 15 for the other samples. To capture the repeat expansion of the sína and ORF72 gene, we performed PCR analysis on the 10 DNA samples which were derived from blood, as this method was also sensitive to fragmented DNA. The patients for whom we had DNA were not closely related, so we set up a filtering strategy where we prioritized non-synonymous variants in exonic regions and splice site regions. And we also filtered on allele frequency thresholds from the NOMAD study. And we filtered against Faroese controls. And using these varying filters, we screened 58 ALS risk genes for rare and potentially pathogenic variants. Illustrated here on the right, one can appreciate the variants that have been discovered over the time, as also discovery methods have been improved, especially with the sequencing. But we also performed a genome-wide search using more stringent criteria to identify rare heterozygous variants, rare homozygous variants, and also try to look for variants shared between cases. So we estimated the pathogenicity of the variants by using CAT scores. We also looked at expression data and literature to discern possible function of the gene. We found that one patient carried the C9 ORF72 expansion, and this patient had family with also ALS, as can be seen in the pedigree here. However, we did not have genetic material on these patients and could not know how the expansion would segregate in this family. The other major ALS genes like FOS, TAR, DPP, and SOD1 did not seem to be causes of ALS in the Faroes, and neither did the other minor genes identified. In our genome-wide search, we discovered many rare variants, of course, but I would like to point out two, as these were shared between the ALS patients to a larger degree than many of the other variants. And in the gene INPP4A, a variant was identified, estimated to be likely deleterious. And this variant was shared between 4 patients with ALS, as can be observed on the plot here in orange. We did not know the specific effect of this variant, but looking at data, it seems that one function may be that it protects neurons from exototoxic cell death, and that is a mechanism that has been related to ALS. So another variant was found in VPS13A. This variant was, however, predicted to be tolerated, but it was identified in 5 patients, illustrated here in green, and 5 patients which seem to cluster within patients from Føroya. Those are the cases on the lower half of the plot. The protein product of this gene may be involved in lipid homeostasis in the nervous system, and also several mutations have been found in this gene and also other homologous genes, VPS genes, which associated with neurological disorders and development disorders. Yeah, so to sum up, the incidence of ALS was relatively high in the Faroe Islands during 2010 and 2020, and a geographic cluster was observed in Føroya. Similarly, the frequency of familial ALS was somewhat higher in the Faroe Islands. We found that the sína and ORF72 expansion was causal of ALS in the Faroes, although it was only identified in 1 person. And other commonly mutated genes didn't seem likely causal in the Faroes. However, we did identify rare variants in 2 genes. So these will need to be studied further for a potential function. And next, it is about Alzheimer's disease. Alzheimer's disease is the most common form of dementia. It is also a neurodegenerative disease caused by neuronal death, which leads to cognitive impairment. And as with ALS, there currently is no effective treatment to stop or slow the progression of ALS, so it is a disease which you ultimately die from. The incidence of dementia has been estimated in the Faroe Islands before, and the crude incidence was found to be 5.1 per 1,000 during 2010 and 17. Compared to other European countries, the incidence of dementia in the Faroes seemed to be within the lower range as compared to Denmark, where it was 10.5. Exactly how Alzheimer's disease is triggered is not known, but many risk factors are known to associate with the disease. Age is the largest risk factor known, and also various lifestyle factors have been associated with Alzheimer's disease, these being smoking, diabetes, high blood pressure, and high cholesterol. And also, genetics and heredity is a risk factor for Alzheimer's disease. In a little less than 5% of cases, mutations in 3 genes, APP, PSEN1, and PSEN2, cause rare forms of early onset familial Alzheimer's disease. But for most of Alzheimer's disease cases, the cause is multifactorial, where also risk genes are involved. The strongest genetic risk factor for Alzheimer's disease is the E4 allele of ApoE, and it is relatively common in the population, as can be seen in the figure here. Approximately 14% of the general population have 1 variant, and carrying 1 E4 allele increases risk of Alzheimer's disease approximately 3-fold. Approximately 4% carry 2, and then a risk will be increased approximately 10-fold. But many other risk loci also associate with Alzheimer's disease, and currently GWAS studies have been successful in identifying approximately 70 genes associated with the Alzheimer's disease. However, all of these loci or risk variants captured by GWAS only explain a small proportion of risk. And then researchers have developed various methods to circumvent this, and polygenic risk scores aggregate risk variants to capture a greater proportion of risk than the individual variant can do. And polygenic risk scores are based on summary statistics derived from GWASs, and a person's genetic risk can be calculated by summing up the number of risk allele variance this person has and multiplying by the effect size or the estimated effect size of that variant. And very simply put, every person will receive a number which reflects their genetic risk. So in a population, most people will have neutral risk. Some will have low risk for disease, while others will have a high risk of a disease. In the Faroese population, we knew little about the genetic underpinnings of Alzheimer's disease. And in this last study on Alzheimer's disease, the aim was to assess the predictive ability of a polygenic risk scores in relation to Alzheimer's disease in a Faroese case control study. And for this purpose, we used participants from the Faroese Alzheimer's cohort, which is a nationwide study launched in 2005. And data presented in this paper were from September 1st, 2015 to October 9th, 2020. From the two largest GWASs on that time point on Alzheimer's disease, we selected 49 genome-wide significant SNPs, and these were genotyped in the Faroese case control study. We performed single SNP association tests of 39 of the SNPs that cohered with quality control procedures, and also built 2 polygenic risk scores. And we built 2 risk scores because we knew that the effect of ApoE is so great, so we wanted to see what the effect was with ApoE, but also see if all the other SNPs were able to capture some risk. And model performance of the polygenic risk scores were assessed using area under the ROC curve. Compared to controls, patients with Alzheimer's disease were 3 times more likely to harbor an E4 ApoE allele. So ApoE was also a risk factor for Alzheimer's disease in the Faroe Islands. And of the 39 SNPs that we tested for an association, only 1 was significantly associated after correcting for multiple testing, and this was the E4 risk allele. The performance of the polygenic risk scores, again, was assessed using area under the ROC curves, and this is a way to show how good the model is able to discriminate between cases and controls. And for the polygenic risk score, including ApoE, the model had an AUC of 80.3%, which is quite high. So in 80% of the times it could discriminate between cases and controls. AUC was a little smaller when excluding ApoE, but it was 63 and still had some predictive ability. So to conclude, it seemed that single SNP associations were quite difficult to identify in a case control setting, and this most likely relates to the small sample size and variants. Some will be common, but as GWASs increase, you will get more and more rare SNPs. So then it will also be rare in the Faroe Islands, and in effect can be quite difficult to identify. And also we observed that 10% of the SNPs that were genotyped were monomorphic in the Faroe Islands, so we have a little less variability in the population. But in aggregate, polygenic risk scores had good discriminatory power, and this was despite of the small sample sizes, and despite of the fewer variable sites, and also despite differences in minor allele frequency and possible differences in LD patterns. So with that, I want to thank you all for listening and acknowledge all the help I have had through this time. Of course, my supervisors, Maria, Tolben, and Annas, but also all the other collaborators. And thanks to the dissertation committee for letting me have this presentation. And thank you to all the funding bodies who have funded these studies. Yeah, thank you.
Yes. Thank you, Malan, for this excellent presentation of your work. Very interesting. And now, the first examiner, Professor Orla Hardiman, will ask questions and comments, and she should be with us on Zoom. So maybe we'll see her here in a while. Yeah. Welcome to you, Orla.
Thank you, and good afternoon.
Good afternoon. You can see us and hear us.
I can see myself twice now, which is...
You can hear us. That's the most important.
I can hear you.
Yeah.
I won't see if I'm upsetting the poor candidate, because I can just see myself.
Okay. But
Hi, Orla.
I will give you the word to you, Orla, now. Please.
Well, first of all, thank you very much. First of all, for giving us the privilege of reading your thesis, which I really enjoyed, and also for your very executed presentation today. I think you captured all of the main features of your findings, and I think it was both an excellent thesis and an excellent presentation. The other thing I would like to commend you on is your excellent command of English. It can't be easy performing this sort of work in a language that's not your native language, and I think we who are native English speakers are always very impressed with the quality of the command of language, both written and also spoken. So I'd like to commend you on that as well. Your thesis is very well written in English and reads better than many pieces that I've read from native English speakers. So congratulations on that as well. So I have to ask you some mean questions. I think that is my job. But I'm not going to be very mean, because I couldn't think of any really, really mean questions to ask you because your presentation and your thesis is very good. So I'm going to ask you some just slightly annoying questions or comments. So I really liked the work. I think looking at populations that are restricted is really very interesting, because we know that ALS is a complex genetic disease, and that the risks are mediated partly by our genetic makeup, and we know that different populations, for that reason, have different genetic risks. So I think the idea of studying this in the Faroe Islands, which is a population isolate, is excellent. Of course, the limitation there is that your population is very small. So I'm going to just make some comments on that in general. But to start off with, because I'm a neurologist, and because neurologists tend to be kind of detail-oriented people, I would just like to pick you up on one comment you made at the very beginning, which is the nature of ALS, which you say is effectively two types of neurons, the upper and the lower motor neuron. Of course, that's true when we examine people, but there are, of course, many other cell types that are involved, or even neuronal populations that are also involved in ALS. And I'm just wondering if you thought any part about that, if that's something that you have a view, because I'm going to develop that point in a few minutes as well, in terms of the way that you were able to examine your population. So would you like to expand a little bit on the characteristics of ALS and our views now about ALS as being a disorder primarily of upper and lower motor neurons, and whether you'd like to expand any more on that point?
Yeah, so if I understand the question correctly, do you want to talk about the cell types, or is it
not so much the cell types. It's more the idea that that we were always taught in medical school that ALS is a disorder of upper and lower motor neurons, and that is how we diagnose it. But we know now that there's a great deal more involved in ALS, and particularly, I think it's in your thesis, the non-motor components of ALS, and particularly the cognitive and behavioral components, which of course are not motor neurons at all.
No, of course, So I guess I have been working only with ALS patients, but during this progression, as I have worked with this population and of course looked at literature, I have come to appreciate that it isn't only a motor neuron disease, because it has clinical overlap with, for example, Ph.d, which is usually classified as a kind of dementia. So there's some interplay with, the frontal lobes. But also there are probably other brain centers involved. As I understand, for example, it overlaps with psychiatric diseases. So I guess that the disease isn't just a motor neuron disease, but there are many other players in relation to the disease also that probably should be taken into account when looking at these patients.
I think that's absolutely correct. And of course, we know now that the mechanism of disease is not just the motor neurons either, but the microglia and the astrocytes are involved as well. But I think it's difficult because clinically the disease is still characterized by the presence of upper and lower motor neuron degeneration. And in fact, many places still classify ALS as a neuromuscular disease. So I was very pleased that you had characterized it as a neurodegenerative disease, because I think that really is what it is. It's part of the neurodegenerative syndrome.
So I thought that ALS was a complex disease, but that just added another layer of complexity.
So this the reason why I'm suggesting this is because some of the work that you did, and I think again, I think your thesis is very good, and I think there were limitations in what you could do by virtue of the nature of the design of your thesis. But one of the things that I thought would be worth discussing was, first of all, the classification of ALS, by the Ellis scoring criteria, because we've moved a little bit away from that to criteria about whether it's the Gold Coast criteria. So it's a much simpler classification system. But also, you referred to the Byrne criteria, which is actually my group, Susan Byrne, who defined a structure for determining whether somebody's familial ALS or not. And one of the things that struck me in your thesis was whether that is excessively rigid in the Faroese population, possible, probable, and definite. The second point that I'd like to expand on that, but also on if we accept that there are overlaps or relationships between ALS risk and other disorders, whether it's correct to only include ALS as the determinant of whether somebody has familial disease or not. So when Susan was undertaking that study, we included frontotemporal dementia as well, but not other disorders. If we looked at this again in the Irish population, and we looked at other family history of ALS and Ph.d, but we also looked at psychosis. And that increased the rates of familial ALS if you expand out the different relations, the different diseases that may share a genetic basis. So I just would like you to expand a little bit on the decision to use a relatively stringent possible, probable, or definite familial ALS, and whether you might have considered thinking about including Ph.d, if possible, and even possibly other psychiatric disorders, particularly psychoses, to help you to define the rate of familial disease. So we've written a bit about this. Marie Ryan also published some work on this. So just expand a little bit on
Yeah.
that. Criticism, really, it's a comment.
Yeah, yeah, of course, and that is something I've been become aware of as I have progressed in my studies. But the data that I came to was already collected, and not much had been done on ALS before. So I think the decision to maybe try to identify the frequency of familial ALS was because we kind of wanted to lay some groundwork to be able to compare us with other populations and make the argument for why this population was special and why we needed to do the genetic research. However, if I had expanded and included FTD cases, our argument would probably have been even more strong. So I definitely think that we should include FTD cases, and that is something that we can do. The incidence study on Alzheimer's disease also included, I think, was it from 2010 to 17 or something, at least 11 FTD cases. And so we know they are there, and we could, of course, try to identify them and link them through pedigrees to our already existing data, and probably do the same for other phenotypes also. And as you say, it is very limited with the few ALS cases that we have, so we would have a stronger argument if we were able to find these large families with phenotypes that probably overlap, and then probably could look at the genetics.
So that's another question that I had, and it's really a question about the way that the health records in the Faroe Islands are collected, and whether it's possible, like it is, for example, in Sweden, to identify individuals from a kindred point of view back. It seems like you have very good records. Whether you would be able to reconstruct pedigrees based on analysis of records, genealogy records. They can do this in the Netherlands as well. And in Scandinavia, it's more difficult to that in Ireland. But is that possible to do? Would it be possible to construct very large pedigrees from your genealogical data in the Faroe Islands?
Yeah, so that would definitely be possible because, well, we have excellent genealogical data, and they're also quite easily accessible as they are electronic. And we also would be able to identify the patients showing whatever phenotype we were interested in. So I guess that would be possible to make these large pedigrees with
So is it possible to link the genealogical data with health records data? Do you have health records taken back over a number of generations where you could link the genealogical data with the health records data? Is that possible?
Yeah, so that would be possible. I haven't really tried to extract medical data, but that is possible. We have electronic health records, but otherwise, I guess you can look for them in paper form. So maybe it would take some work, but it is definitely possible.
One of the things that you could do then would be a sort of an identity by descent analysis, and to see if there are clustering of kindreds with particular clusters of clinical phenotypes that link less with FTD and maybe other diseases as well. So it looks like a good population in which you could do that, I think.
I think so.
One of the points that you made in your thesis, and you mentioned it in your talk as well, was the rate of sporadic disease, the rate of familial disease being 5 to 10%, and the sporadic disease, for which you did mention it's a complex genetic disorder. So the heritability studies that have been performed, they've been performed using twin studies, GWAS studies, and then using population-based studies as well. So that actually works out at about 51%. So genomic risk accounts for about 50% of the risk, even in non-familial ALS. If you take out C9, it goes down a little bit. But again, the Faroe Islands would be a great place to study this. And that's really what you were doing, the Alzheimer's study, was looking at heritability risk when you're looking at the genomic profiling. So it would be nice to be able to do that in ALS as well, I think, with the studies that you have. I want to move on to your study on incidence, and one of the things that really struck me, because we've been looking at this in the Irish Register as well, there are two things that struck me in this which I think are very interesting. One is that the incidence went up in your cohorts, in your two different cohorts, and that was partly related to the age of the population, I think. But I noticed that the age of onset moved to the right. The peak age of onset in both males and females shifted to the right. So the peak age of onset in the first cohort was somewhere in the mid-50s, and the peak age of onset in the second cohort was somewhere in the mid-60s. Would you like to expand a little bit on that?
Yeah, so of course we saw an increase in incidence, and I have speculated why. So is it random? That could be the case since we have a small population and there is uncertainty when sampling. But of course, I've also been speculating, may there be some improved ascertainment? Maybe previously, older people could maybe not have gotten the diagnosis. So if we have become better to ascertain ALS at the older age, maybe that's why we see that the age has increased. I think that could be a possibility.
So the peak was in the 60s, so that would be, I think you showed a peak age rather than it wasn't the peak age.
It's become sorry.
Yeah. So it moved to the right. No, it's really 67. Interesting. Yes, it's a really interesting finding, and we've seen that in other registers as well. And I wonder, is it to do with the age being a risk factor, but also healthy aging being a risk factor? So the age of onset moves a little bit to the right as the population is healthier for longer. It's a really interesting finding.
Yeah, of course, yeah.
Yeah. So another question I have for you, and I know you can't answer it, but I think it's a comment really, is that you were very reliant on a single neurologist or a group of neurologists who had been around for a very long time, who had been making the diagnosis of ALS. But of course, our understanding of the disease has changed over time as well. And it may be that some we have biases, of course, and all of us have biases as we develop. Do you think that's a limitation in your study, that there was a single point of ascertainment? And was there any mechanism of doing recapture within your data set to try and limit that bias? And sometimes it's not difficult; it's difficult to do that. But I'm just wondering, did you consider it in your method?
No, so we didn't really do any recapture or address that possibility. Of course, it could be...
Sometimes it's not possible because the data sets are...
Yeah, it is really small also. Of course, there could be some bias. However, I think that even though he has practiced as neurologist in the Faroe Islands, most of the cases have also been sent to Denmark for their diagnosis, and also neurologists have been here in the Faroe Islands to look at the patients. So, I'm quite confident in in that.
Do you think it might have changed though? Again, in our register, we've noticed changes with, we call it sort of information creep, that what we would have considered as a relatively rigid definition of the disease maybe 20 years ago, we might be a little bit more open to including a slightly less rigid definition. For example, the presence of significant cognitive impairment and maybe some mild bulbar dysfunction. We would probably classify that now as ALS, whereas in the past we may not have done so. So,
of course.
One of the potential reasons for the for the change could be a slight change in how we would classify the disease, even though it would still fall within the Ella score classification. It's still very much a clinical evaluation, and so some of the change in instance may relate to a change in both the evolution of the clinical prowess of the neurologist and also a recognition of a wider range of conditions that we might accept as being ALS, whereas we mightn't have in the past.
Yeah, of course, that could be a reason. I understand that the diagnostic criteria have become way more lenient in recent times. So of course there is the possibility that more patients are being identified as having ALS. That could be an explanation. You're right.
I think it's very hard to know, but it's just something I think that should be considered.
Yeah.
And that finally, I had a lot of sympathy for you on your genomic studies.
Thank you.
It's hard to make any conclusions from the 10 samples that you had.
Yeah.
And then, the 20 samples, but 10 really of good quality.
Yeah.
Just remind me, how many of those were familial or seem to have any kind of familial component to them of those 20 samples?
Sorry?
Of those 20 samples, how many were familial?
Familial?
Family history?
4 of them had a family history of ALS, but none of the familiar cases were included, like 2 from the same family. So I only had the one.
You only had one person who...
Yeah, from a family.
And would it be possible to do any sort of identity by descent with the samples that you had to see if they were cryptically related? I know everybody in the Faroe Islands is cryptically related. I understand that. So that would be a confounder. So that would be difficult to do, I think, given the small numbers as well.
What? Sorry? What did you say?
To determine whether the people who had ALS were more cryptically related than the general Faroe Island population. I think you did that in your thesis, and I think they weren't. Is that correct? To determine whether there was a greater degree of relatedness between the people who had ALS compared to the rest of the people in the Faroe Islands.
No, using the pedigree data, it didn't seem.
Could you do that genomically?
Yes, of course, if include
greater degree of runs of homozygosity, or it's called cryptic relatedness.
Yes.
Identity by descent to determine if these people were closer relations to one another than your normal Faroe Island population.
Yeah. So that is probably something that we should have included. Have controls that we could do that with, so we could compare the cases with controls.
But it's difficult because the population is so such an isolate that it might not provide any additional information, especially from the Southern Ireland. They might be more related to one another anyway. There were all my comments actually. I think there's one other question actually I have for you about relating to the age of onset. Did the life expectancy in the Faroe Islands change during the 2 periods of study?
Oh, I'll just find it here. If the life expectancy changed?
Of the overall population in the Faroe Islands over the 2 intervals of study. Did the life expectancy in the Faroe Islands go up?
It seemed to go a little down.
Okay.
Survival time, yeah.
No, not for the patients, but for the overall population.
Oh, for the overall population, yeah, sorry. Yeah, it did go a little bit up, the survival time. Or the
overall survival rate of the population.
Yeah, it did.
Okay, that's. I think you've answered my questions really well. I think, as I said, it was an excellent presentation. I think I asked you some unpleasant and mean questions, and I think you answered them really well. So thank you very much for that. It was a really nice and insightful presentation and a nice insightful interview. So thank you very much.
Thank you, Ola. This was pleasant. Thank you.
Thank you very much for the questions and comments, Orla. And now we will have a coffee break for about 20 minutes. So 20 past 2, we'll be back. Yes? Thank you. Welcome back. We now will continue, and now the second examiner, Jørgen Nielsen, will ask his questions and comments. I don't know if we have Jørgen with us on Zoom. Yeah.
I should be there.
Yeah. Welcome, Jørgen.
Thank you very much.
And I give the word to you.
Thank you. Thank you, Malan, first of all, for this very nice work. Despite the relatively small size of the different data sets, it has also been a very nice presentation until now. So I will now, as Ola did so very well, try to be mean in a pleasant way. And I would like to start on the front page of your thesis. It's about ALS and Alzheimer's disease. And why that? What is the commonalities between ALS and AD? Wouldn't it have been more nice just to concentrate on ALS and skip AD? Wouldn't it be more coherent, or what is the reason to do that?
Hi, Jan.
Hi.
So you think why I work with ALS and Alzheimer's disease?
Yes, why
Yeah.
also Alzheimer's disease?
So, to be honest,
Yes.
yeah. So I was interested in working with genetics. That was my primary research interest, and came upon this study, study population of Alzheimer's disease, ALS. So and started to work with this, and as I mentioned, the data wasn't so complete. So I thought that I would expand on my studies on the methods part by including also the polygenic risk scores in relation to Alzheimer's disease. So it's maybe more of a methods correlation than a phenotype correlation, I think.
Yeah, I agree. Yeah, okay. Then I would like you to go to the abbreviations list. Could you tell me what is the intention of such a list?
Well, the intention of an abbreviations list is to, well, when you use some of these long words to short them down here and to be able to go back. And
Yes.
Okay, so you think
perfectly, but is that especially easy? Because when you find some kind of abbreviation, as for instance the first one, A52 residue and so on, UBA52, then I would go back and look for UBA52 and then have the full word after that. Wouldn't that be natural?
yes, I see it now.
Yeah.
Yeah,
I found it a bit difficult.
yeah, I think you're right. Yeah.
Okay, no problem. Then for the introduction, I would also like you to expand a little of on neurodegenerative diseases as such, and what do you think? How do you define neurodegenerative diseases?
Yeah.
Explain a little more detailed.
Yeah, so I don't know how detailed I can be, but neurodegenerative diseases, well, as the word implies, it would be diseases, I would say, in the brain where various neuron types die or degenerate. So
I agree, but you write in the first line 3 that it is also in the peripheral nervous system. So it's a progressive loss of nerves in the central and peripheral nervous system.
yeah,
But is it the same amount of neurodegenerative diseases in the central nervous system as in the peripheral nervous system? And you cite Federica Perrone in this reference number 1, and state that this paper defines neurodegenerative diseases as progressive loss in both the central and peripheral nervous system, but in fact that is not true. They write that it is primarily affecting neurons of the central nervous system. So I think it is a free interpretation of this.
Yeah, okay, yeah.
Citation.
Yeah.
Okay, then I would like you to go to paper 1 and ask you, were there any surprises in your results?
So, yes. And no, because I'm new to research on neurodegenerative diseases and Alzheimer's disease. That's a new field for me. But just going through literature, I was maybe surprised that maybe bulbar onset was quite high in the Faroe Islands. But I don't know if it's something else that you are thinking about.
No, I'm thinking about all those islands of the Faroe Islands. There's also one called Streymoy.
Yeah.
Yes, and the prevalence on Streymoy.
Yeah. I'll just have a look.
Would you make a comment on this?
Just wait a minute.
I think this short report is only about Suðuroya.
Here.
But
so
you not mention Streymoy? I think
so there was a
very interesting.
Yeah, so there was a decreased incidence, a prevalent or occurrence, I should maybe say, of diseases in Streymoy.
Yes.
Yeah, so I didn't really talk a lot about that.
No. Could that be used in some way? Could you think about a new research project concentrating on Streymoy?
Yeah, that's a good question.
Yes.
Where the frequency of disease is quite lower.
And why that?
Yeah, that's a good question.
Yes.
So
could there be any protecting factors or something?
Yes, there could, but I know in relation to Alzheimer's disease, where also protective factors have been identified. I think I haven't thought in that ways in relation to Alzheimer's disease. I think I just thought about the high impact variants that cause the disease, but of course there can be protective factors also.
Yes, do you know some genetic protective factors in inherited neurodegenerative disorders caused by a special kind of mutation? Have you ever?
Well, I guess I have come upon several without really
I think so.
Remembering, but of course in relation to Alzheimer's disease, there is the...
It's in relation to cancer, that is what I'm...
In relation to cancer, protective variants in relation to cancer?
Yes.
Yeah, maybe.
In neurodegenerative disease. Yes, CAG repeat disorders, polyglutamine diseases.
Okay.
If you harbor one of those mutations in the Huntington gene or in SCAR1, 2, 3, 6, 7, 17 and DRPLA, you are at a certain level protected against cancer. And
Yeah,
it's very
okay.
robust.
Yeah,
So maybe
I didn't know that. But,
okay. Yes. Then paper 3, I would like to ask whether this manuscript is submitted by now and
yeah, so
what is the status?
yeah, actually it is not. So when we performed this study, which was in 2020, these were the samples that we can use.
Yes.
And it was quite limited. So I think we're at a state where we think that maybe we should elaborate a little bit on this study. There have been new cases of ALS, so should we perhaps include these? And now, after talking to you and you and Ola, and you have revised the thesis. So thinking about if one should perhaps include Ph.d. cases, and so yeah, it hasn't really been published yet. We're in a thinking box, I think.
Yeah. That's all right.
Yeah.
Okay. Then I would like you to comment on the list of genes. It's table, the supplementary table S1. Page 72.
Yeah.
And you call it the ALS-associated genes. What do you mean by that?
Yeah, just a minute. Yeah, so maybe it should just be. Well, I meant that it is genes that some may be associated with ALS, but some probably also increase risk of other diseases. So this could be a candidate list, maybe, instead of ALS-associated genes.
Yeah. So they are not all causal
No.
of ALS? No. If we take this SPAST gene, SPAST, do you know the most common phenotype of this gene?
Actually, I don't. No, not of that.
No. I think that the reports associating SPAST mutations with ALS are really dubious. Normally, it gives a late onset spastic paraplegia, but not as ALS with this fast progression. So I think it's not really a ALS-associated gene, but you can always discuss that.
Yeah.
And how did you make this list?
Well, I did have inspiration when going through various databases. I didn't really know at first where to look, so I found inspiration in another study who had looked at these variants in relation to ALS in a population isolate. So I thought I would do the same in the Faroese population.
Yeah, that's good. Could you also comment on using exome data on those 206 Faroese individuals sampled as part of a study on autism, right? As normal control data?
Yeah, so I had these ALS patients, and that was what I could get sequenced. I didn't really have any whole genome sequencing controls, but for the identified variants, I did want to see how what the frequency was in the Faroe Islands also. So it wasn't really control data selected in relation to ALS, but a means and readily available data to maybe filter a little bit against.
Okay. Did they have autism, or were they controlled?
No, they were
controls. They were controls in this autism study.
Yes.
Okay. Good. You also write somewhere that you did a sex check based on X chromosome heterozygosity rate. Why did you do that?
Why?
Yeah.
Yeah, because, performing a sequencing study, that was new to me, so I have, of course, looked around to see what everyone else does, and I just wanted to make sure that the sex concurred between my samples and the genetic data. So I guess that was why.
That was why. Did you consider before what to do if you found out that one of the samples didn't have the sex which the individual thought they had?
No.
How to convey this information?
Well, I didn't. No, I really didn't consider that. But given that they have all been deceased, I'm not sure I would bring the information back to them. So.
No. And how did you convey the results of this gene results to the individuals or the families?
Well, information hasn't been brought back in person.
So the families, they do not know that they harbor a inherited disease, or?
Well, many of them have been looked at in, for example, Denmark. Some will have given consent to look at this, but doing a genetic analysis on a deceased, then we are that we are allowed to do. But of course, if identifying any high impact variants, the question is whether maybe we should consider going back with the data.
Yeah,
information.
Yeah. You didn't set up a small committee beforehand to handle.
No, that's maybe something one should have done.
Sometimes it's done.
Yeah.
And it may be a good idea because you never know what you will find.
Yeah.
So, yes. You also used this algorithm expansion hunter.
Yeah.
What was your impression of this tool?
Well, I had help with this part, and my impression wasn't that good. I think I don't remember what the sequencing depth should be for success, but I don't think our samples were good enough for using expansion hunter. In those individuals from which we had blood, expansion hunter performed a little better, but we had the positive control, the person with the C9 or 72 expansion, and that wasn't really called by expansion hunter.
No, I saw that. So
there is some uncertainty, I guess.
Yes.
But
it would be nice if we could call those expansions.
Yeah, but I guess it doesn't really do that 100%. So
no, it doesn't. Okay. Why did you check for expansion in the ATX1 and 2 genes? What is the rationale for that?
Well, in general, I guess, I think it seems that expansions, it has been recognized that for many of these neurodegenerative diseases, expansions do play quite a significant role. And in some instances, there is overlap. So I think it made sense to at least look at some of these in relation to ALS. Since we were on expansion hunter anyhow, it made sense to look at these variants at least, even though data wasn't perfect.
No, but I agree. The ataxia 2 intermediate expansion is one of the most robust genetic risk factors in sporadic ALS, so I think it's a very good idea to look for those.
Yeah.
And then you write a lot of this INPP4A and VPS13A genes.
Yeah.
Do you believe that your findings are real, that those are associated with ALS, or what is your opinion?
Well, I believe
and how could you get closer?
Yeah, so I believe the findings are real, but given that people from Føroya most probably are more related with each other compared to the rest of the country, the question is if if these variants just segregate within the population of Føroya, or if they actually do have something to do with ALS, that I'm not sure. But of course, that is something, and I tried to look at expression data and looked at literature to try to discern some function and argument why they could be related to ALS, but I don't know. And that should, of course, be investigated further. And you asked, of course, if we had published a paper, and maybe before we do that, we should verify that the findings that we have are actually findings.
Yeah, I think that would be nice. I think this VPS13A could be really relevant. It's involved in mitophagy, and other ALS genes are involved in autophagy and other processes linked also to mitophagy. So I think that would be really relevant. I don't know about the INPP4A because that's cerebellar and striatal expression. And is that relevant for ALS? I think that's more dubious, but maybe.
Yeah.
Okay, Malan. Then how would you in an ideal world bring your research further from here?
Yeah, that's a good question.
Yeah.
Well, I guess money is always a limitation. So in an ideal world, I would probably do some more case finding. I would sequence more patients, maybe also controls. I would look at maybe the other phenotypes related to Alzheimer's disease. And in relation to repeat variants, I don't know, maybe do some specific sequencing of these to see if they really are present in the Faroese population. Yeah, but that's a lot. There's a lot of things you could do, but of course,
a lot of things to do.
One is so limited by sample size. So I guess maybe in the future one could go further with this as sample sizes would increase.
Yes. But I also think that you could bring it further by doing some functional analysis. And, what are in fact your plans? Where are you now, and what are your future plans?
I want to continue with performing genetic studies here in the Faroese population, because I think that is something that should be done. So maybe in relation to progress or continue the search for ALS genes in the Faroes, and also work with other neurodegenerative diseases. I think that could be very relevant for me to continue this path. Yeah.
I hope you will.
Yeah.
I think I have no further questions. Thank you so much for letting me be opponent on this very interesting work.
Thank you for agreeing.
Welcome.
Okay, bye.
Bye.
Yes, thank you, Jürgen, for very good questions, and thank you, Malan. And now there's an opportunity for you here in the auditorium to ask questions, or if you have any comments, please just raise your hand and I will come with the microphone. Yes? No questions? Everything is very clear?
Yeah.
Okay, then I will step outside and have a short meeting with the other in the assessment committee, and I will come back as soon as possible, and then we will have the final recommendation. Thank you. Can you hear me now? Yeah. Great. Thank you. And Orla and Jürgens is also with us? Yes, thank you. So I will now read up the final recommendation from the assessment committee. The PhD candidate, Malan Johansen, has by her dissertation, Epidemiology and Genetics of the Amyotrophic, Lateral, Sclerosis and Alzheimer disease in the Faroe Islands, demonstrated knowledge of and the ability to use epidemiologic method and standard bioinformatic methodologies in research of international PhD standard. By public defense, Malan has further elaborated on this and demonstrated her capacity to discuss and reflect on the points raised by the evaluators in a fully satisfactory manner. The members of the assessment committee find that Malan Johansen, by her dissertation and public defense, has documented the ability to carry out a research project by independent application of scientific theory and methods, and in that way her work has contributed to advancing research at a level meeting international standard. So in conclusion, the members of the assessment committee find the dissertation and the public defense to be of sufficient academic standard for award of the PhD degree. Congratulations. I will hand you the diploma to look at. And give an applause. Congratulations. And now I will thank you all for being here today. And now we are going to celebrate with a reception. And please stay and have some drink and food. Thank you very much, and thank you, Malan, and congratulations.